about more, especially when we do chromosomal microarray, about these particular terms. So you should be aware about them. So these two initial lectures are basically just to revise and get you to know about some of these terms which we will be using during our analysis. So if we are meant to be two-way, any of the lectures, please stop in between or at the end, whatever you prefer. But at that same time, do ask questions. And as we have begun, we will go from basics then to the advanced. So let us have everyone on the same page. That is why this initial one or two lectures we kept to bring everyone on the same, and from the next lecture onwards it will be going more detailed into fish. Okay. So when we talk of these different fields, so we have different names that pop up. So the whole genetics per se started with this field of human genetics, that is in the early 60s and 70s. And that time genetics was mainly studied either by the plant geneticists or by the population geneticists, and we really did not have any human utility of genetics. So it was more of a research, and that time it was called as a field of human genetics, where population geneticists were studying large populations for polymorphisms, for different frequencies of the polymorphisms in populations. However, with the advent of karyotyping, that was the first genetic test which could be applied to the patients. That is when the specialty of clinical genetics came up, where people now started identifying syndromes, the cause of those syndromes, the cause of those genetic diseases, and then doing prenatal diagnosis, genetic counseling. So that's the field which started of clinical genetics, where you apply the genetics into clinical field. And then we had medical genetics, which combines these, both the clinical genetics as well as the laboratory genetics. And now with the advent of predictive medicine and personalized medicine, we call it as the genetic medicine or genomic medicine, where again now we have fields of pharmacogenetics and predictive medicine that come up. So it has slowly evolved over the ages. And if you look at the scope of medical genetics, what all different things come, then it includes the baby in utero, where fetal medicine doctors will look at the baby in the uterus using ultrasonography. If the fetus is terminated because of some abnormality, then fetal autopsy is used to identify more malformations. And then to know the genetic cause of these abnormalities, we either use the chromosomal analysis, that is the clinical cytogenetics. We can use molecular genetic studies, which look at the level of DNA, or we use the biochemical genetic studies, where we look at the level of protein, either as hormones, enzymes, when we test. So those are all the laboratory genetic tests that are employed. And then we have the personalized medicine and predictive medicine, where we try to identify people at risk of certain diseases, or people at risk of developing complications to the drugs, or they are more susceptible to certain drugs or not. So that is the whole scope of medical genetics that we have. We had a nice lecture now on chromosome. So however, in the last lecture what we saw was a very broad or a very low resolution picture. If you look at each of these bands, it is 5 million base pairs, that is 50 lakh ATGCs. Unless more than 50 lakh ATGCs are affected, you won't see anything on the chromosome. So it's a, although it's a whole genome picture, but it's a very low resolution picture. In the next techniques, that is FISH, we are going to take that resolution from 5 million base pair to maybe 1 million base pair or even lesser. Okay, so from 5 million we come down to 1 million, and then when we go to microarray, we'll take that resolution even lesser to few kilobases, maybe 50, 100 kilobases. So it's higher and higher resolution, at the same time higher and higher cost that these tests will incur. So let us see some of the terms or the principles which especially we'll be using when we talk about the microarrays. So what's the difference between an allele and a locus? Allele is one of the two versions of a gene. We all know we have two chromosomes for each pair. So each of these chromosomes will have different forms of the same particular gene. So the two forms of the gene on two different chromosomes, these are called alleles. So here capital A and small a, these are two alleles on these two homologous chromosomes. Locus is the position of the gene on the chromosome. So position of that particular, so on chromosome 1 on the P arm, if you say, it's the geographical location. So when we talk about locus, it is the geographical location. When we talk of allele, it is the two forms of the gene on the chromosomes. So an individual inherits two alleles, one from father, one from mother. If these two alleles are exactly same, like here, capital A or small a, we will call it as homozygous. If the two alleles are different, we call it heterozygous. Okay, so these terms are very clear to you: allele, locus, homozygous, heterozygous. Next is, what is a genotype, what is a phenotype. So genotype refers to the individual's total collection of genes. So all genes in an individual will consider it genotype. So the two alleles inherited for a particular gene will be the genotype at that locus. Okay, so genotype at this particular locus is capital W, capital W. Genotype at this locus is small w, small w. The phenotype is the observable trait, what we can observe by our eyes or by imaging, such as height, eye color, blood type. So that is the phenotype, that is what we observe. And trait is the specific. So the phenotype comes from the trait, that is the specific characteristic of the organism. So we always try to match the genotype with the phenotype in any of our testing that we do. Okay. Next, coming to gene, what's the anatomy of a gene? Very important to know the anatomy of a gene, because we are going to look for abnormalities of these genes in the copy number variations. So gene, it's an ordered sequence of nucleotides at a particular locus, okay, on a particular chromosome, and it results in a gene product which could be either a protein or a RNA. Okay, there are some genes which only code for RNA, they don't form any protein, others form proteins. So the anatomy of the gene is that the gene starts from the promoter. Before promoter, there are other sequences called as enhancers or silencers, which are long distance regulators of the gene. But ultimately, the transcription starts with the promoter. After promoter, you have, this is something called as a transcription start site. Okay, so here the transcription starts, and this is the translation start site. So transcription is from DNA to RNA, translation is from RNA to protein. So here we have the transcription start site, here we have the translation start site. The sequences between these two are called as the 5' untranslated region or 5' UTR. Then we have this translation start site, where there is the first coding region, the first amino acid is methionine. Then you will have exon, intron, exon, intron. Then you have a translation stop, that is the stop codon. And then you have a transcription stop site. The sequences between translation stop codon and transcription stop site is the 3' untranslated region. This whole structure is what we term as a gene. So these individual genes are then regulated by multiple different other enhancers and silencers. Important to remember of another entity called as pseudogenes. Okay, so these are genes which are very similar to the genes. It is thought that they have come about because of duplications throughout evolution. The gene has got duplicated, however it has accumulated certain mutations because of which it is inactive, it is not producing any protein. It is very similar to the gene, but it is not resulting in any protein, but it creates a lot of problems while our interpretation of either NGS or microarray analysis. So you have to remember about this particular entity. The next is the two types of complementary sequences. So when we talk of complementary sequences, we have one is the complementary sequence, that is we have the golden rule, A pairs with T, G pairs with C. So these are called as complementary nucleotide sequences. On the other hand, complementary DNA or cDNA refers to the DNA that is synthesized from the messenger RNA. cDNA is not present in our cells anywhere. cDNA is what we synthesize in the lab. Okay. So, but it helps us because it is DNA, complementary is RNA, complementary is cDNA. So cDNA has exact same sequence like the DNA. That is why we use cDNA when we number any mutation, because that number remains same in the cDNA and the DNA. Whereas RNA, it is a reverse complement. So if there is T in the DNA, it becomes U in the RNA or A in the RNA. Okay, so now there is confusion which we call a mutation, the RNA mutation or the DNA mutation. So for our ease, we always use the cDNA numbering for saying any mutation site. Okay, but it is not present in the body. It is synthesized in the laboratory or in silico you can synthesize. If there is G in the DNA, it will form C in the RNA. Again, it will be G in the cDNA. So your DNA and cDNA is same sequence. Okay. Coming to the next term, what is a polymorphism? Polymorphism is again two alleles which are present in the population, but these alleles are present so common that we say they are not likely to be associated with any disease. So if alternate allele is present in more than 1% of population, we call it as a polymorphism. Polymorphisms are variations which make us all different from each other but do not lead to any disease. So you can have either a single nucleotide polymorphism, just a single nucleotide change, or you can have repeat polymorphism. So the CAG, number of repeats, or you can have a copy number variation polymorphism. Okay, so single nucleotide polymorphism or triplet repeat polymorphism or copy number repeat polymorphism. So what is this copy number variation? The whole idea of doing microarray is to look for copy number variation. So copy number variation is basically submicroscopic deletions or duplications. That means they cannot be visualized by microscope. So that's why we call them submicroscopic. What is microscopic can be seen by doing a fish. What we cannot see in microscope, those are the copy number variations. Usually we call a copy number variation when there is deletion or duplication of more than 1000 bases. That is 1 kb. Okay. So anything less than 1 kb, if it is a 500 base pair or a 600 base pair, that is also a copy number variation. But these terms are used because a 500 or a 50 base pair deletion or duplication we can easily identify by doing a Sanger sequencing. Okay, Sanger sequencing typically you can do up to 1 kb at a time. But if there is a 2 kb deletion, now you can't see it by Sanger sequencing. So you need a microarray for that. So that is why this term of copy number variation is called for deletion duplication which is more than 1 kb. It may be 2 kb, it may be 200 kb, it might be 2 MB also. So whatever any deletion duplication more than 1 kb, we call it as a copy number variation. These are mainly detected by microarray, and these again can be benign copy number variants or pathogenic copy number variants. Okay. So a deletion and duplication can be benign without causing any disease, or it may result in a disease. So when it results in disease, we call it pathogenic. Otherwise, they are called as benign normal variants or polymorphisms. Okay. So let us go on to now understanding what are the spectrum of disorders that we see in humans. So if you put on a line the etiology of disease for all human disorders. It could be either a totally genetic etiology or a totally environmental etiology. So what are some totally genetic etiology conditions? Yeah, monogenic disorders like sickle cell anemia or beta thalassemia or any other than monogenic disorders, chromosomal disorders. Okay, so we'll have chromosomal disorders or monogenic disorders which are totally genetic. They, if the mutation or the chromosomal abnormality present, disease present, abnormality absent, disease absent. So they don't need any environmental effect to cause the disease. What are totally environmental conditions?
Exposure.
Yeah,
Infectious disease.
infections will come to infections. Are they totally environmental? There is a doubt on that. What are totally environmental?
Communication.
That also may not be totally environmental.
UV radiation, all things.
Maybe motor vehicle accidents, maybe a lightning strike.
Yeah.
Yeah. So, or maybe nutritional deficiencies, maybe scurvy,
Metabolic.
maybe very. These are totally environmental. You don't need a genetic predisposition. Okay. And then there are diseases in between, like coronary artery disease, cancer, diabetes, asthma. These are called as multifactorial disorders or polygenic disorders or complex disorders. They have a genetic predisposition and an environmental factor. When both come together, then only disease will happen. If only genetic predisposition is there and favorable environment is not there, disease will not occur. Only environment is there but the genetic factors are not there, disease will not happen. That is why you see certain very obese people, they don't have diabetes. And then you see certain very thin people, but they have diabetes. Okay? So it's not necessary that only genetics or only environment will play a role. It's a combination of both. And that is why these diseases, we understand very less about their genetics, and that is why we have very less from the therapeutic side. We only can control these diseases. Whereas here we understand much more about these diseases, and that is why for some of these diseases we are even able to cure them, because we understand their pathophysiology, why they will happen. Whereas here we still are not able to understand. Now where will you put infections or allergies? They will come somewhere here, not really totally environmental, and this we have seen in our COVID pandemic. Okay, so all the virus was same, but it was affecting different severity in different individuals. So infections, allergies, they also have a genetic predisposition, how they will respond to a particular infectious agent. Okay, so let us see about the genetic disorders. If you have to classify genetic disorders, they can either be chromosomal disorders, we heard about them in the previous lecture. The others are monogenic disorders, single gene disorders, also called as Mendelian disorders. And the third are the multifactorial or the polygenic or the complex disorders. So sickle cell disease, classic example of a monogenic condition where you have a single gene, that is the beta globin gene, a single mutation and a single disease. Okay, this is the classical paradigm, but this is not the case in every monogenic disorder. As we know, there are multiple genes which cause the same disease. There are multiple mutations in same gene causing the same disease. There are multiple diseases caused due to mutations in the same gene. So you have all different types of combinations and complexities. However, this will be what we will call it as a classical Mendelian disorder. What about the multifactorial disorders? Why do we call them genetic if they are not totally inherited? So what is seen in multifactorial disorders is that they aggregate in families but don't segregate in Mendelian fashion. Okay. So what is aggregation and segregation? Segregation means it follows the Mendelian rules of allelic segregation during meiosis, whereas aggregation in families means if you have a family with diabetes, you will have multiple people affected with diabetes, but you can't have a recurrence risk. You cannot give a specific recurrence risk. So there is some aggregation, but there is no rule, kind of like a Mendelian disease segregation. Multiple genes are involved, multiple environmental factors are involved. So that's the complexity. So although these are common problems in community, in public health, but we know much less about them. And what is the reason is that compared to monogenic disorder where it is single gene, this is multiple different mutations in different genes. Impact of variation again is different among different families. Okay, so in one family, one gene may be major contributor. In another family, another gene may be major contributor. So that adds further to the complexity. Although we say diabetes itself is a collection of multiple types of diabetes, which we still don't understand, because each one will have one particular gene which is contributing more to the disease in that family. As opposed to that, the monogenic disorders are much more easy to understand, and the microarray also will be coming across these monogenic disorders. So let's see, because we will be talking more in terms of pedigrees when we analyze the cases in microarray. So this is a typical pedigree that we will draw. So it has the two parents joined by the relationship line. Then we have the line of descent. Then this is the relationship line with each individual having its own line. So here the husband and wife have 4 children. Squares are males, circles are females, 2 daughters, 2 sons. The shaded ones are the affected ones. Okay, so that is the principle of drawing a pedigree. When you shade a individual, that person is affected. So here you can see AB and BC are the alleles. Okay, and what you can make out here, the A allele is associated with the disease. Okay, so in this way you can make out through the pedigree. So let us see some different types of inheritance patterns, which we will be again coming across when we do a microarray analysis. You can have different types of inheritance patterns leading to the disease. So this is a typical autosomal dominant inheritance pedigree. What are the characteristics here? You can see both males and females are equally likely to be affected. Affected individuals are related through another affected individual. Once there is no disease here, it is not transmitted to the next generation. Male to male, male to female transmission is seen. This is a vertical kind of transmission. So these are all the characteristics, and classical examples are achondroplasia, Marfan syndrome. Almost all copy number variations which are de novo, they are all autosomal dominant in inheritance. Okay, some of the copy number variations in microarray you will find both parent and child will have the same copy number variation. So that is also a autosomal dominant kind of inheritance. As opposed to this, the autosomal recessive inheritance is where both parents are unaffected, but there might be consanguinity, and then multiple children in the same generation will be affected of both genders. And typically the examples are like thalassemia, cystic fibrosis, or spinal muscular atrophy. The third important type of inheritance is X-linked recessive. Again, copy number variations can show X-linked recessive inheritance, where mothers will be carriers and only the males are affected. Females are not affected because they have 2 X chromosomes. So this is a crisscross kind of inheritance. The dominant is a vertical, the recessive is a horizontal inheritance. This is a crisscross inheritance. Male to male transmission is not seen because male gives Y chromosome to his sons. Only female carriers transmit to male children. So Duchenne muscular dystrophy or hemophilia, these are examples of X-linked recessive inheritance. So what we know about traditional genetics is there were always people who worked in laboratories, and then there were people who worked with bioinformatics on our computers. However, in today's world, with the large amount of data coming either from a microarray or a next generation sequencing, you have to have some knowledge of both the sides, otherwise you will not be able to practice this kind of a medicine. So today's clinician, today's researcher has to be well aware of the bioinformatic principles, and the same thing you will be seeing when we go to analyze the microarray. These chips have millions of dots on that. Manually it is not possible to analyze this data, so you have to have some bioinformatic analysis to be able to understand what this biology means, or what this patient is having that particular disease. To understand that disease, you need to have good hold over the bioinformatic analysis. The more you are able to do, the more you are able to analyze, but a basic understanding has to be there. So we will see how even in your own clinics, or when you do the analysis, how you can make use of these bioinformatic tools to understand what is happening in the patient. Okay, so that was a brief overview for you to know different terms that we may use during our discussion on microarray analysis, or when we do the exercises for doing the microarray analysis. So any questions on these? Are very simple kind of, it's basically a revision of what you might have already read, to go ahead further now, because henceforth we will be talking more and more detail about fish and the microarray network and analysis. So any questions till now?
Sir, what is the full form of RBS in that structure of the gene? Sir, you have shown that slide.
What is that?
Structure of gene.
Ribosomal RBS and the gene structure.
RBS. What is the full form of RBS?
RBS, I also don't
It's
know. What is
ribosomal
that?
binding site.
Ribosomal binding site. So maybe the translation initiation, so it must be ribosomal, but the protein from RNA to protein happens on ribosomes. So this is the site where it binds to the ribosome. So this is basically the translation start site. It is the methionine here. Always it will be ATG or AUG. Okay, so that AUG will be, they call it the RBS, maybe the ribosomal binding site. So how many of you have done microarrays, either done or seen the results, or have some idea about microarray? Yeah, you have seen results of microarray from a laboratory or something? And about fish, how many of you have, you're doing fish, or you have heard of it? You have heard. Okay. So we'll go on to the next one. So now we'll